Before I start my presentation, I just want to give a very brief introduction about New York University's global network. So we have 3 degree granting campuses located at New York, Abu Dhabi, and Shanghai in China. And then we have 11 other academic centers throughout the world, right from Accra, Ghana to Tel Aviv, Israel. And this is our beautiful campus located in Abu Dhabi, United Arab Emirates. And we are located in an island called Saadiyat Island. And we are right there. And this is our campus. And we just celebrated 10 years of completing our mission here in Abu Dhabi. Just a brief statistics about the students here. There are 1,600 plus undergrads, 100 plus graduate students. And this is one of the most diverse campuses around the world. So we have 115 plus countries represented, people speaking 90, 100 plus languages within a small campus. So if any of you are interested in visiting NYU Abu Dhabi, I'd be happy to welcome you in, provided all the COVID restrictions are not there. So this is briefly about what we do in the Vijay Lab here at NYU Abu Dhabi. We work on 3D printing and bioprinting for tissue engineering, regenerative medicine, drug testing, and medical devices. So predominantly, I work on 3 areas: 3D printing and bioprinting, some of computational studies to deal with complex 3D structures, and then 3D printed medical devices, which includes implants and prosthetics. So these are some of the works that we do in the lab, right from nerve guide conduits that you see here on the top row, on multi-layered skin scaffold, meniscal scaffold, tendon repair. And then, on the bioprinting side, we do work on neural bioprinting and skin bioprinting. And then on the implant side, we work with architected metamaterial structures like triply periodic minimal surface based scaffolds. Just to set the stage, I hope you guys have heard about 3D printing before. It is the process of producing a 3D object directly from a CAD model, layer upon layer, as opposed to other manufacturing methodologies such as subtractive or formative manufacturing processes. So it's also called rapid prototyping or additive manufacturing. And one key point to note here is additive manufacturing is an umbrella term. There are more than 40 different processes which can be grouped under one manufacturing methodology called additive manufacturing. So there are several different additive manufacturing processes which can print different categories of materials, including metals, ceramics, polymers, hydrogels, and composites. So the figure, the video that you see on the screen, it's something fascinating called food printing, right? So it is the chocolate being printed on a piece of bread, right? So 3D printing has been used right from printing hard, robust materials such as metals, right up to cultured meat and chocolate and cookies, etc. So like I said, additive manufacturing is an umbrella term. So the ASTM F42 Committee on Additive Manufacturing grouped all these different processes into 7 different categories, as you can see in the slide. I'm not going into each of these types and explaining, but just to let you know that any additive manufacturing process could be categorized into one of these 7 groups. Now that we know what is 3D printing, what is 3D bioprinting? If you incorporate viable living materials or cells into the printing material, then the process is called bioprinting. So there are 3 main or commonly used processes in bioprinting. The first one is laser-based. The second one is inkjet-based, which is very similar to the printers that you use every day in your office. And then the third and most commonly used bioprinting method is the extrusion-based printing, where the material is forced out of the nozzle using pneumatic, piston, or screw-based methods. So before we move into the research that we do in the lab, it's important to understand why a three-dimensional structure is required for the cells to grow, right? The native human tissue is three-dimensional. People have been doing cell culture, especially all the life scientists. They do their cell cultures on two-dimensional substrates, well plates or petri dishes, etc. But then you could actually see a lot of difference in the cell behavior when you culture them on a 2D substrate and a 3D environment. So the cells have more natural cell shape when it is cultured in a 3D environment, more natural interactions between cells, a multi-layer cell sheet is formed, improved stem cell expansion, and cells behave more like those in vivo. In short, the three-dimensional environment gives a home environment for the cells to attach, grow, proliferate, and differentiate. Now, why do we need 3D printing or bioprinting? Two main reasons. One is the growing organ shortage, and then replacing animal testing. So there are many different medical applications of additive manufacturing, right? Starting from educational models where you can use 3D printing to print anatomical models for student education or patient education. And second, surgical models where you can print patient-specific anatomical models for either preoperative planning, surgical simulation, or even designing and printing of customized surgical tools or guides. And the third application would be medical devices, which includes wearable devices, prosthetics, and implants. And finally, tissue engineering, where you can print tissue engineering scaffolds on which the cells can be seeded and matured in a bioreactor, or you can directly print tissues using bioprinting, which then could be used for regenerative medicine, drug testing, and precision medicine applications. So in my talk, I would try to briefly cover one example each from implants, scaffolds, and bioprinted tissues that we do in the lab here. So the first example or the first application of additive manufacturing in implants, we work on some of the bone implant designs. So to give you a brief background, your hip could be damaged due to many reasons. It could be aging related, like osteoarthritis, or it could be autoimmune disease, like rheumatoid arthritis, or it could be other reasons like post-traumatic arthritis, avascular necrosis, or childhood hip disease. So the gold standard is a hip replacement surgery. And what you see here is a standard hip implant that is currently available in the market. So there are, of course, many challenges related with the current hip implants, but I would like to highlight only two important needs or challenges of the current hip implants. Number one, the hip implant now that is currently used has a much higher mechanical property compared to the native bone. So during the normal physiological activities, the implant takes most of the load, preventing or shielding the native bone, the native surrounding bone, from experiencing any stress. So this results in reduction of bone density, and it would result in the removal of the implant after some time, implant loosening, and then you would have a secondary surgery and so on. And the second need or challenge would be the lack of biomimicry in the implants, right? The porous metallic implants currently in use do not mimic the structural and functional architecture of the native bone. So what we hypothesize is that given the limited choice of implant materials, basically the implant materials are made of metals, stainless steel 316L or titanium alloy, which is Ti6Al4V. right? So there is already a limited choice in terms of implant materials. So the challenge lies in implant design. How do you design the implant so that you could improve the biomimicry and reduce the stress shielding effect? So that's where we think architected metamaterials would come to rescue. So we work with triply periodic minimal surfaces, and being porous and possessing high surface area to volume ratio, we hypothesize that these triply periodic minimal surface based designs would help in better osseointegration, or in other words, a better integration with the surrounding native bone. So these are some of the TPMS designs that we work with. So the first one is a primitive unit cell. The second one is a gyroid, and the third one is a diamond-based TPMS structure. So as you can see from the graphs here, you can play around with the unit cell geometry, the relative density, and the properties of the base metal, and you can get a variety of mechanical properties to suit the biomimicry of the native bone. So for example, if you take a typical bone, there would be a hard outer bone called the cortical bone, and then the core of the bone would be a spongy or tubercular bone. So each of these regions have a different porosity, different pore size, and of course, different mechanical properties. These are anisotropic. So we can use the TPMS-based unit cells and structures to design such a gradient scaffold or a functionally gradient implant. So I've given an example here where you have a radial functionally graded scaffold based on TPMS with two distinct regions with different porosities and different properties. We have also printed them using several different materials. The example that I show here on the slide is with alumina, which is a ceramic material, and the process used for printing these is SLA, stereolithography apparatus. And again, with the experiments that we did, the mechanical testing, we showed that the mechanical properties can be varied by playing around with the unit cell size, thickness, and the other parameters of the TPMS-based designs. So an integrated approach of combining the right method of design, which is TPMS-based or functionally graded in this case, a right choice of material, say ceramic or titanium or 316L stainless steel, and the right process, the kind of 3D printing that you use, could lead to novel customized bone implant designs, which would mitigate the stress shielding effects caused by the conventional implants, and also enhanced biomimicry. So future biological validation with in vitro and in vivo cell culture studies, which we are currently doing in the lab, would further the clinical translation of this approach. The second example, or the application of additive manufacturing that I want to talk about is the tissue engineering scaffold. So we use a specific 3D printing method called electrohydrodynamic jet 3D printing. So we apply a very high voltage between the nozzle and the substrate to get a very fine fiber. We could go down to a 5 micron diameter fiber. This is a much improved and much better system than electrospinning, where you cannot control the macro and micro morphology of the scaffolds together, but in this process, we could control both the macro and micro morphology of these fibers. So we have used this EHD jet 3D printing to print scaffolds for various tissue engineering applications. As you can see here, neural, skin, tendon, meniscal, esophageal tissues, but I would just talk about the neural tissue applications in this talk. So let's understand what is the nerve guide conduit first before moving on to the work that we did in the lab. The peripheral nerves in the body can sustain injuries due to loss of structure and or function of peripheral nerves. And the cause could be an accident, trauma, or any other cause. which leads to partial or complete loss of sensory, motor, and autonomic functions and neuropathic pain. And what would be the treatment method? The gold standard that is followed now is nerve autografting, right? Taking the nerve from some other part of the body and then repairing this particular peripheral nerve. But there are many challenges associated with nerve autografting, which includes donor site morbidity and limited availability. So that's where the nerve guide conduits come into play. And these NGCs are increasingly being considered as a potential alternative to nerve autografts. So as you can see from the figure here, these NGCs act as a bridge between the severed nerves, which would have cues to help in axonal regeneration or neural regeneration. Now, there are many requirements of an ideal nerve guide conduit, right? Of which conductivity is one of the desired property. Why? Because bioelectricity plays an integral role in maintaining normal biological functions, which includes signaling of the nervous system, functioning of the muscular system, etc. And by having a conductive scaffold or a conductive NGC, you can provide external electrical stimulation to the cells. And by providing this external electrical stimulation, the cellular activities such as cell adhesion, migration, DNA synthesis, protein secretion, etc., could be modulated, thereby having an effect on differentiation and tissue regeneration. And in the stem cell therapy, if we are able to control the cellular activities using external electrical stimulation, then it could act as a check on undirected and uncontrolled differentiation, which is the greatest challenge or risk associated with stem cell therapy. So there are many limitations of conductive scaffolds or substrates that are found in the literature. One, the conductive polymers have weak mechanical properties and hence could not be printed into a 3D structure. And that's why most of the works that are found in the literature are 2D conductive substrates, and the conductive polymers mostly are non-biodegradable. So to address these challenges and overcome the research gap, we printed what is called a conductive biodegradable nerve guide conduit. So we used a block copolymer of polypyrrole and polycaprolactone along with pure polycaprolactone, right? And we varied the concentrations of PPY BPCL, and then we fabricated the scaffolds using electrohydrodynamic jet 3D printing. And as you can see from the graphs here, we were able to print a nice structure with controlled porosity using this process. The pore size were in the range of 100 to 150 microns. And the conductivity is in the range of millisiemens per centimeter, and it increases with increasing concentration of PPY BPCL. And with the addition of PPY BPCL to the pure PCL, we can see that the mechanical properties decrease drastically. And whatever we have achieved, it mimics the native human nerve properties, which is the ultimate tensile strength of 6.5 megapascals. And we have done extensive biological studies on it. Briefly, we cultured human embryonic stem cell derived neural crest stem cells on these scaffolds, and we have differentiated them into peripheral neurons, right? So as you can see from the figure A here, the cell proliferation on the conductive scaffolds, the PCL-PPY scaffolds with 3 different concentrations, are much better than the non-conductive pure PCL scaffold. And we have done the differentiation studies as well. And then what we could see is we could witness a higher gene expression of peripherin, PRPH, and neurofilament heavy chain on PCL-PPY scaffolds, which shows that there is a stronger maturation of the stem cells to peripheral neuronal cells on the conductive scaffolds than on a non-conductive scaffold. So these are again some of the protein expressions using immunofluorescence staining, beta-3 tubulin, neurofilament heavy chain, and Dhabi. again showing that the conductive scaffolds actually help in a much better neuronal differentiation than a non-conductive scaffold. The last application that I would like to talk about is on bioprinting, right? So I talked about the implant, I talked about a scaffold, which is nerve guide conduit, and then the third application that I'm talking about is a 3D bioprinted human skin. So printing of tissues using bioprinting method. So we do print different kinds of tissues, mainly neural and skin. And in this talk, I would just talk about the skin bioprinting project. So as you might already know, skin is the largest organ of the body, accounting for about 15% of the total body weight, right? And then it has a very complex multi-layer structure, as you can see from the figure here, which makes bioprinting interesting, right? As I already told, this is a layer by layer printing process. And because the skin is a layered, complex multi-layered structure, bioprinting comes in handy to print these kind of tissues. So we have developed a comprehensive process to print human 3D skin, right? I'm not going into the specifics of the process as such. It is already published and the paper is available. You could get the link from my website, right? So briefly, what we did is we combined the electrohydrodynamic jet printing and extrusion bioprinting system. So the first step, we have a 3D printed PCL mesh. PCL is polycaprolactone, right? And then we have an acellular base layer printed out of collagen, right? And then we go to printing of the dermal layer. So we mix the fibroblasts in collagen and then we bioprint them on top of the acellular base layer. And then after incubation for 2 to 3 days, we go on to print the keratinocytes, which forms the outer layer or the epidermis of the skin. And then we do the air-liquid interface lifting to get the differentiated skin. And then we do a detailed biological characterization. So this is a video of printing of the PCL scaffolds. Right, and this is again another video of printing of collagen-based hydrogel. Right, so this image that you see here is a bioprinted skin made out of human cells. It is a living skin construct, and we could see that up to 21 days, we could maintain this printed structure in culture. Which is more than enough to do some of the drug or cosmetic testing. And one important observation here is by combining the EHD jet printing and extrusion bioprinting, right, we have prevented the shrinkage of collagen. So just by having a PCL mesh at the bottom, it actually prevented the shrinkage of collagen, which is one of the greatest challenges faced by skin scientists, right? So we have done extensive characterization of the printed skin, which I'm not going to talk in detail about, but briefly what we could say is our bioprinted skin could actually meet the standards of the 3D organoid culture, which was done manually in the labs around the world. So finally, the last 2 minutes, I want to touch upon the vascularized tissue bioprinting. So now that we have developed a method for printing the skin, we want to also try having a vascularized skin construct, right? So we have optimized the bio ink for printing of vascularized tissue, which is xanthan gum and collagen based bio ink, right? This is the design of the printed construct. So we have fibroblasts as the 2 outer layers, and then we print endothelial cells in the 2 inner layers. So we have 4 layers of bioprinting. And the post-bioprinting characterization proved that you could see the microvasculature, capillary-like structures blood vessels forming in the bioprinted construct. So these are the H&E staining, which also proves the same. So we were able to print vascularized tissues using bioprinting. So with that, I would like to conclude again by showing this slide that 3D printing and bioprinting are a high potential techniques with which you can address many of the medical needs or gaps that are present in the system now. And we specifically work on medical devices and tissue engineering, where I showed an example of an implant, a bone implant, based on TPMS-based design to reduce the stress shielding, to mitigate the stress shielding effect, and to enhance the biomimicry. And I've also shared an example of 3D printed scaffolds, which is electrohydrodynamic jet 3D printed nerve guide conduits for peripheral nerve repair. And then finally, we also saw an example of bioprinted tissues, which is a human skin bioprinting, and a part of which was a bioprinting of a vascularized skin construct. And thank you for your patient listening. And this is my email. Feel free to contact me for any questions or further collaborations. And I would be happy to answer any questions that you have now.